Hello, my name is John Kearney, and today I am going to bring you into another episode of Into the Lore Tower. So, as I've already told you guys before, I have a biology degree from the University of Scranton, and today I'm going to talk to you guys a little bit about what I know from those days in biology. So what really inspired this video today was when I was watching Matthias DeStefano as he was speaking at the Gaia Conference Center. I believe it was the Immersion Conference a few years back. I'll look into exactly what year that was, but he was talking about his interpretation of how he understands the universe in relationship to the biological system of our body. And he said the words from my memory, "If you want to understand the secrets of the universe, then understand exactly how biology works first." He even had this whiteboard up that showed all the different body systems, and he related it to the zodiac systems, and it was really cool, honestly. So that's why today I want to discuss what I know in biology and try to relate it into everyday terms the way that I do at work through eyeballs. My patients say I'm pretty good at it, but I guess we'll see what I can do today, right? All right, so without further ado, I think we'll start with a little bit of a history lesson first. So, before genes were even discovered, a very famous Dutch chemist named John Baptist van Helmont, who you can see pictured here, had a recipe in the 17th century to make a mouse. According to the recipe, you just had to take one shirt soiled in sweat, one sheath of wheat, you leave it in the sun for 21 days, and bingo, you have spontaneously generated mice. Now, this is the same man that established mnemonic chemistry, which is just another way of saying working with gases. So being the man that discovered carbon dioxide, he does know his stuff, and it's not his fault that he was limited by the beliefs of his time. His time had its basis back all the way in Aristotle's days of 1350 BC, as he stated in his work on the generation of animals. In this work, he talked about his belief of epigenesis, which was foundational to the previous spontaneous generation argument I just mentioned. Aristotle performed a series of observational and philosophical arguments against the belief of preformationism, where a fully formed human, otherwise known as a homunculus, existed inside the seed or egg already. So basically the premise here is, instead of having something that develops into the fully formed human body, the human body is already fully formed in just this really teeny tiny version. And then as it develops, it grows in size. Now I don't know if you've looked at a baby recently, but I'm a little confused where that came from in the first place. But at least I could see where Full Metal Alchemists got their inspiration for the homunculus for. Since Aristotle's logic was the scientific foundation for medieval and then later Renaissance teachings, it took the invention of the microscope to finally move humanity away from this line of thinking of either epigenesis or preformationism. So, we get to thank a man by the name of Robert Hooke and his usage of the compound microscope in 1665. He was able to see what he called tiny rooms that he called cells. He documented these findings among several other observations in his work, the Micrographia. This work was clear documentation that life formed in repeating patterns, not as random masses of matter. Hooke's work also served as inspiration for others to pick up the microscope. This is an example of the microscope that Robert Hooke used to make his first observations of the cells. You can see that it looks very similar to a compound microscope that we use today, but I really like the pattern that's on his. I would love to get a replica of this thing because it looks sick. You could also see an example of the different lenses and spheres that he had on the left side here. This is a more modern depiction of some of the photographs that Robert Hooke has in the Micrographia. You can see that he was able to see these repeating patterns of tissue from perpendicular cross sections of bark and parallel bark surfaces. He also had some hand drawings of the cross section of what he called pipes and the longitudinal sections of them as well. So this is if you're looking at like them from the top down, and this is if you're looking at them turned 90 degrees, looking through the pipe. But in 1674, someone by the name of Antony von Leeuwenhoek used a simpler, more powerful microscope to observe living, moving cells. Instead of using multiple lenses, like in the compound microscope we just saw, this one used a tiny glass bead. You can see the objective lens here. The specimen would be put in front of this pin right on top there, and you can see that the observer would look through this tiny glass bead that allowed him to magnify up to 200 to 300 times, which I'm not gonna lie, for a little tiny thing, that's pretty good. He's credited as the first to observe bacteria from his own dental plaque. He saw amoebas in pond water, and he also called these things living animalcules. So you can kind of hear the predecessor to molecule coming out here in animalcules. Here's some modern photographs of some of the observations that Leeuwenhoek would have made through his little simple microscope. And here's a better physical representation of that microscope that he used. All right, now that we've had a little bit of history of how cells were discovered, I think it's important that we kind of have a little discussion on why cells are so important in the first place. Because observing these things clearly were important, but I'm also not trying to bore you guys with a biology class, because we've all taken this in high school. I know we all have some foreknowledge with this. I really don't know what you've retained. I'm not trying to assume anything, so I'm just saying exactly what's pertinent to my discussion today. And if you guys have any questions about things further, I can always point you in the right direction, or you could check out the source that I'm using for this discussion today, because I found this really sweet website called Lab Exchange, which is really sweet. So Lab Exchange is a free resource that I found in my research here that has a library of over 34,400 resources that anybody could check out. What I'm using to help my discussion today is what's called pathways. And you can see that they have a bunch of different advanced level ones. But if you go over here and go to sort by, go to most favorited. Okay. Actually go over here to subject area. And what I did is I clicked on biological sciences. So if you go over here to subject area, you can find some of the more beginner topics to explore. I clicked on microbiology personally, and then I went over here and I went to most favorited. Yeah. So I started scrolling down in like cellular structure and function. That one's perfect, because here's the video that I started this episode with right here. So now we're going to go through a few different examples just to help our discussion today. So thank you, Lab Exchange. I really appreciate your free resources, because it's really helped me in my visualization of this biology that's so microscopic. But first, the reason that cells are so important is because every single living organism is made out of cells. Cells are the basic structure of all life. Structure equals function, and cells group together to help form that function, which starts to form tissues, which starts to form organ systems, which makes up the human body, such as the heart, such as the brain, such as the lungs, etc. So the purpose of even building all of these different systems that have forms and have functions is so that they can work together in order to achieve tasks. And those tasks are directed in the center of each cell by the nucleus. The nucleus is where the DNA is stored, which is where all the encoding information is to help a cell build, repair, replicate, and perform whatever specific duty it has, whether it's a muscle cell, a heart cell, a brain neuron, a spinal cord, etc. But life, as we know, evolves. It took millions of years in order to even get to this point. So, before things became cells that are like ours, they started off as what's called prokaryotic cells. So these prokaryotic cells are lacking the different organelles that we have in our body. They have what's called cell walls, but there's two different kinds of cell walls. And as you'll see here in a moment, that's how we do the different what's called gram staining patterns in order to identify what antibiotics would be effective against these things in modern medicine. But all of our cells, ancestor-wise, started off as something like this. And these are the different shapes that you can see in prokaryotes today. In order to move around, they have these different protein structures called fimbriae, flagellum, pilae. This is their plasma membrane, which is very similar to our own, but something that we don't have, as you saw, is these cell walls. But just like our cells, they have these structures called ribosomes. You can see that right in the middle here is what's called the nucleoid. They don't have a nucleus center like us, and sometimes they have this side DNA strand called plasmids. So all of the cool stuff that you saw in the beginning of this video was an example of a eukaryotic cell, an animal cell, just like we are. And this is a picture that I have on the screen here. As you can tell, there's a dedicated nucleus. And again, I'm not going to go over every single organelle that's in this system, but the important takeaway is the prokaryotes do not have the organelles, while the eukaryotic cells do. Some of the important ones are the endoplasmic reticulum here, because as the nucleus is making DNA, it makes what's called RNA. That RNA is what makes copies of DNA and also makes proteins for the cell to perform the various functions that it needs in order to, again, survive, replicate, live, and perform its duty in the first place. All of these proteins are packaged up in the endoplasmic reticulum here. All these little red dots are what's called ribosomes that move to the Golgi apparatus on the side here. And you can think of the Golgi apparatus like the UPS of the cell. They package up the cells and they deliver them exactly where they need to go. You can see an example of that going on right here, because again, pink, that's how it's color-coded on this exact diagram. It's moving onto the cell membrane to transport some of those proteins out into the extracellular matrix, or to embed new proteins into the cellular membrane, which we'll get into in a minute. Plant cells are considered eukaryotic, and similar to bacteria, they have a cell wall, but the cell wall is commonly called a capsule in bacteria, and that'll become important later on in my discussion, so hang on to that one. This is an example of the cell wall. Now, when it came to modern microscopes, now I believe it took until the 1950s to actually have good microscopes to see the double layer of the cell membrane, but we didn't have the capability to actually see the function and what was going on here to figure out that this is what's called a phospholipid bilayer. So again, without getting into the technicalities behind this, the important points are this side loves water, the inside hates water. That way, not everything can go through this thing, but there are some proteins, and all of these blue structures are proteins that act as channels to help certain molecules get through. But there are also some, as you can see up here as blue and green, glycoprotein, glycolipid. Those act as communication, which I'll talk about later. The cell membrane is able to have a certain level of awareness or detection of its surroundings, and that's how it performs cell-to-cell communication and how it responds to certain environmental signals. This is a zoom in of the nucleus here. So as you can tell, this is a better view of the endoplasmic reticulum and how there's all these different channels for the proteins to go through. And there are these little pores here that allow the protein. This is an example of the DNA unwinding itself, because DNA is bundled up in these X-shaped things called chromosomes, as you can see. And again, that's something that we've all reviewed in biology in high school, so I'm not going to go over that too much. But this is an example of DNA turning into RNA, which is like the blueprints that come from DNA in order to make proteins. So you don't want to take the original copy and ruin it, so you make copies of the original and use those up. Of course, everybody knows the mitochondria, the powerhouse of the cell. Okay, let's get back to the discussion on DNA producing protein. Here's a better 3D representation of a eukaryotic cell. You can see all the little phospholipid heads. Here's the mitochondria in the green that we're going by. You can see the endoplasmic reticulum around the nucleus. We're going through one of the pores. Here's one of the chromosomes that we mentioned, the X-shaped bundles of the DNA. This is the DNA unwinding, and now you can see the phospholipid skeleton with the little nucleotides in the center, and there's your genes. And if you guys remember, there's going to be 4 different bases to these genes. RNA, you could think of that as like the factory where all of this work gets performed. Here come your different bases, and again, each one has a certain pair, and that pair is what makes a protein. There are start codes, there are stop codes. If you want to get more in depth on this, again, you can check out Lab Exchange, and you could look out exactly how this coding works. But a good analogy is to think of it like coding with a computer. Computers are built off of binary sequencing, 0s and 1s. When cells divide, they go from 1 to 2. The code is what drives the function of the computer. Code here is what drives the function of the protein. When it comes to RNA, there are different types. The amino acids are like the little building blocks of the actual proteins themselves. So what's really fun is each little protein, each amino acid building block gets chained together through this process of the RNA. Again, there's a stop code when it's making the exact protein that was ordered by the DNA, and here goes the protein forming into its specific form. So now we're going to go through one of their interactive figures, because I really like this perspective. If you guys want to read the text on your own, feel free, but I'm just going to kind of yap here as I scroll through this thing. But there's this one structure I failed to talk about called the microtubules. In the beginning video, you might have seen the nucleus with all these little things shooting out. Microtubules are like the highways and the supporting structures, kind of like our bones and spine for our body, for the actual cell itself. Proteins make up the cell membrane, like we saw before. As I mentioned with the prokaryotes, they make up these little hair-like things that help the body move. Proteins are also made up of enzymes, which you can think of an enzyme as a catalyst. It helps reactions happen at lower temperatures, happen faster, happen easier. Enzymes just help things happen easier. That's a way to think about it. And the ribosomes we've just been talking about too. So here's a good 3D representation of the cell membrane you saw before. Again, whether these are glycoproteins or glycolipids, don't know, but same story. So as I mentioned before, the cilia are like these little hair-like tendrils that move, but even they're really complex. They're not just made out of one protein, they're made out of a certain bundle. And here you can see these spokes that are shooting out from this center circle. 1, 2, 3, 4, 5, 6, 7, 8, 9. So you can say that's a 9-spoke geometry. But I digress. Everything in the body has a design, and that function has a specific purpose. Again, I just talked about enzymes, but this is how they're able to catalyze these different reactions. You can think of them like lock and key with substrates. There's a little bit more nuance than just that, but for today's discussion, that's all we need to know. And the ribosomes, again, we talked about that in the DNA transcription. So I'm just going to kind of blow through this real quick. But this is a good representation of the different protein structures as they're forming. Because again, these polypeptide chains, those amino acids that were getting chained together, you could also call them peptides. So polypeptides is many peptides. They can form two structures, which is either an alpha helix or a beta sheet. Then there's tertiary structures, as we can see here. And then as these tertiary or third structures that form from the secondary structures start to combine, they act as what's called quaternary structures. And this is a good example of a quaternary structure, because in the blue here, you can see how there's all these alpha helixes, and you can see these beta sheets here. But ATP synthase is one of the driving tools that's used inside the mitochondria to help with cellular respiration. Again, the powerhouse of the cell. Here's just another example of a quaternary protein. This kind of reminds me of like heart valves, if I'm going to be honest, like that meshwork that's going on here. But that's just what my brain is reminded of when I look at that hemoglobin. Here's the different models of how proteins are structured, just in case you see them in different books, different manuscripts, different pictures online. They might be represented in those ways. And here's actually an example of how some diseases aren't necessarily tied to the gene. So the gene could be good in the original copy of the DNA, but as it's being transcribed, let's say the environment is under stress for some reason, that might cause one of these proteins that are normally produced well to get misfolded. And if structure equals function, then that protein is not going to function the way it was intended, if it even functions at all. And if this keeps aggregating is the technical term, this is how some diseases such as Alzheimer's or sickle cell anemia develop in the population. All right, next I'm going to give like a paraphrased version of a presentation given by Bruce Lipton on Gaia called Inner Evolution. And I really like the way that he broke down biology, because he's been researching biology, teaching biology, and he's been talking about epigenetics since at least the 1960s. On Gaia, he was talking about how he was able to put stem cells in 3 different environments in petri dishes, and he was able to get them to grow different types of cells in the body, such as muscle cells, stomach cells, heart cells, etc., etc. And he always talks about describing any biological system like a machine. You would think of it as some type of mechanism. That mechanism has a force. That mechanism either gets turned on and off, and there's a certain feedback. In more simple terms, you could break this down as there's an input, there's a process, and there's an output. Sometimes if a process happens enough times, it could be stored as a memory. That way the output is more predictable or more efficient the next time. Examples throughout the human body that we can find this in is in the human neuron. Again, the dendrites are the input, there's a process, and then the axon outputs to communicate to cells next door. Bruce Lipton has a really good breakdown of what's called the hermetic laws too. One of the laws is called correspondence. You may have heard the words as above, so below. He describes using that as a way to learn about larger biological systems by observing smaller ones, like learning about ecosystems by observing human cells, or learning human cells and applying that to how the human body works, like I did with the microtubules versus the human skeleton, both acting as structural supports. All of those individual nerve axons collectively act as an input in the rods and cones inside of our eye. Light comes in, bounces all the way to the pigment layer on the bottom, travels up through all of these different styles of nerves. They all wire together, they all communicate together, and develop this output that goes right to your brain. And of course, when your brain processes that output, right, it sends a signal straight through your spinal cord. You have a sensory input through all of the different nerves throughout your body. There's a process and an output throughout your spine. So as mentioned before, the brain itself has input in the forebrain, processes in the midbrain, and outputs in the hindbrain back and forth from the spinal cord. But let's think about this for a second. Nerve cells act as our memory, right? So if nerve cells act as our memory, but they're made of the same parts as any other cell in our body, body. Without getting into the science, their cell membrane is what drives the potassium and the sodium to drive what's called the action potential. The action potential is the electrical zap, the actual communication between cell to cell that happens on the surface. And in Bruce Lipton's philosophy, that antenna I mentioned before, acting as a receiver to help the cell process that information and react with a certain output. In this case, let certain ions through an ion-gated channel. This switch acts as the cell's perception. It acts as awareness of the elements of the environment through physical sensation. So, in biology, things evolve. We started from prokaryotic cells, we moved to eukaryotic cells, but how exactly did we get there? Does survival of the fittest explain that? If we want to talk about that more, we can do that down below. But Bruce Lipton's theory is, in order to gain more intelligence, in order to gain more evolution, you need to have more surface area on your cell membrane. So this is his theory as to why, going back to the prokaryotes, they have capsules. They can't grow any larger than that. The capsule is their barrier. So what do? They start folding their membrane in. Now, let's apply that analogy to the human brain, because the human brain also has a very hard barrier around it, the skull. And what do you know? There is a fold in the brain, similar to the prokaryotic cell on the left, the sulci and the gyri. So, according to Bruce Lipton's theory, you start with organism A, it tries to become the smartest, most intelligent cell to drive evolution, right? These simple colonies turn into integrated colonies. Now you have a new organism. This is how you move from the prokaryotic cells to the eukaryotic cells. This is how evolution has driven all of these cells to collectively work together in order to form these bigger bodies of intelligence in order to further drive its evolution. So as you can see in this graphic here, you go from single cells to bigger bodies that become their own single individuals. And in phase 3, he theorizes that the human community is eventually going to drive itself into humanity. All right, so thank you for listening to my little yap session on my understanding of biology. Again, I went to the University of Scranton. That's where I learned all of these things about biology. I currently work in healthcare at Northeastern Eye Institute. I've been there for about the last 8-9 years now, because I think I just had my anniversary. But all of this biology helps my understanding of my story writing for SPLICER. Because, as you can see on my screen here, Jamin's holding one of Lady Helen's vitality capsules, right? So when a SPLICER agent is injured, when they take damage in battle, they are able to regain their vitality after taking one of these vitality capsules. So my inspiration behind this is, since mana is the energy that drives life in the SPLICER universe, Lady Helen was able to develop this technology to supercharge the mitochondria. That way, every single cell in the body can produce more mana, which helps this lead towards this healing effect, this cellular repair, dare I say. But thinking about other series that I've watched in the past, I could definitely relate this connection theory or evolution through cooperation that Bruce Lipton was talking about to a series like Naruto, for instance, because the dynamic between Naruto versus Sasuke was always the power of friendship, the power of gaining a collective through your merit, through your kindness, through your good deeds, versus having singular power, being by yourself and scorning all of those around you at that cost. Just like in their final battle, Sasuke was ready to give up Naruto for more power, but it was that friendship and that connection that Naruto had that led to all of the power he had in that final battle. And in that final arc in general, if I think about it, Naruto always talks about how chakra is the connection that binds everybody together. And there's definitely more of a discussion we can have on the Rasengan and the spiral, but I think I'm going to save that for the sacred geometry lesson that I want to do, not next episode, but probably on the 4th one. So what I have planned coming next is, again, as above, so below, applying smaller systems in order to understand larger systems. Next, I'm going to bring you guys into my understanding of plasma physics, the electric universe theory, and a couple new things technology-wise that I've seen floating around on YouTube that are helping me when it comes to writing things such as the Guardians and the overarching framework that is Yggdrasil itself. So that'll probably be episode 3. So yeah, I think I'll do the sacred geometry in episode 4, because sacred geometry is a really good tool to help understand form equals function. But I think this is where I'm going to call it today. So if you guys wouldn't mind, go to Global Comics right here. You can check out SPLICER. All of our chapters are currently free. We started this journey back in 2021. We released our last chapter in August of 2025. So If you want to catch up to the series, if you haven't read it yet, here's your chance. If you've read everything already, let's talk about it. But I had fun talking about this, and I hope you had fun too.